So good afternoon everybody. Today we are starting with the third unit, Recombinant DNA Technology, or also you call it as genetic engineering, or you also call it as RDNA technology. So what do you understand by recombinant DNA? Usually when the DNA is taken from two different sources and they are joined together and expressed in a particular organism. Means what? दोन डिफरंट सोर्सेस मधून दोन डिफरंट ऑर्गॅनिझम मधून DNA काढलाय and then you have combined or ligated together, joined together, and then that DNA, which is called as recombinant one or the recombinant DNA, you are introducing into a suitable host cell and allowing that recombinant DNA to undergo cloning or multiplication or to undergo a transcription and translation process. So that is what is called as the recombinant DNA technology. Okay. So this has slide changed?
No.
So recombinant DNA or gene cloning, you can call it as gene cloning or you can call it as genetic engineering. So recombinant DNA, it is a form of artificial DNA that is created by combining two or more sequences of DNA, which usually it is not found or normally it is not found in that particular organism, and this you have produced artificially by joining together or by process of gene splicing. So recombinant DNA technology is a technology which allows the DNA to be produced via artificial means, and this procedure has been used to change the DNA in living organisms and has played an important role in the medical as well as the pharmaceutical industry. So we have many future aspects. So this recombinant DNA technology has gained importance nowadays in the field of science. So if you observe this picture, what do you observe? It is a Chinese dragon, and usually the Chinese dragon is a organism which is formed by joining together the different parts of different animals, joined together by different parts of animals. So this dragon, it is a mythical means जे काल्पनिक म्हणतात आपण, काल्पनिक क्रिएचर आहे. and this creature, it can fly, it can walk, and it can मीन्स तो तोंडातून फायर पण बाहेर काढू शकतो. सो सच टाइप ऑफ मिथिकल क्रिएचर, इट इज सेड टू बी हॅव्हिंग अ डिवाइन पॉवर. सो इट कॅन समन द विंड, इट कॅन समन द रेन, कारण त्याच्याकडे आग आहे, त्याच्यामुळे तो विंडला संपवू शकतो, रेनला संपवू शकतो. And this dragon is said to be made up of many different types of animals on the earth. Similarly, if you observe in our Hinduism, you have Lord Ganesha. This is also a recombinant creature, which it is also a mythical organism, which has got the head from the elephant and the whole body part from the human being. But it is we call it as a divine power or divine or God. So this creature, the dragon creature, it is made up of 9 parts of a Chinese dragon. I mean to say, why this particular organism or particular creature is a recombinant one? Because it has got the DNA from different organism and it has got expressed, and because of this expression it has got a different phenotypic characteristics. So this phenotypic characteristics are usually expressed, and what are those phenotypic characteristics? In this imagination creature, you have the deer's antlers, means सिंग डिअरचे आहेत, कॅमेलचं तोंड आहे, हेड रिजन आहे, हेअर म्हणजे रॅबिटचं आईज आहेत, स्नेक्सची नेक आहे, फिशेस सारखे स्केल्स आहेत, कार्प्सचे स्केल्स आहेत, ईगल्सचे क्लॉस आहेत म्हणजे नख आहेत, टायगरचा पावस आहे म्हणजे त्याचा पंजा आहे आणि ऑक्सचे इयर्स आहेत. सो इट इज अ रिकॉम्बिनंट क्रिएचर. So in this case we mean that recombinant DNA means it is having a DNA from one or two organisms. So we are creating a different chimeric genes. We are creating a chimeric genes when we are splicing or joining together, splicing or joining together the DNA from different sources. So recombinant DNA is cloning. Cloning means you are multiplying several number of time, म्हणजे त्यांच्या कॉपीज नंबर ऑफ टाइम्स आपण बनवतोय, and creation of the chimeric genes. Okay, so recombinant DNA technology is one of the recent advances in biotechnology, and it was developed by two scientists named Boyer and Cohen in 1973. Just I was born in 1973. At that time only the Boyer and Cohen gave this phenomena of recombinant DNA technology. So Stanley Cohen, he received a Nobel Prize in Medicine in 1986 for his discovery of growth factors. And Stanley Cohen and Herbert Boyer, they worked together, and they constructed the first recombinant DNA by using bacterial DNA and the plasmids. Okay, so they took the bacterial DNA or the chromosomal DNA from bacteria, and they took the plasmid from the another bacterial cell, and they made or joined together to form a first recombinant DNA. So what is recombinant DNA now, or what is recombinant DNA technology? Recombinant DNA technology is a technology which allows the DNA to be produced via artificial means. So we in vitro means in the lab, you are joining together, or you are allowing the DNA from two different sources or two different organisms in this technology. The procedure has been used to change the DNA in the living. So when you are joining the two different DNA from two different sources, usually the DNA of that individual organism is getting changed and may have even more practical uses in the future. So in this, it is an area of medical science, this recombinant DNA technology, that is just in beginning to be researched in a concerted efforts. In a concerted means you have to take much more efforts, following all the ethics and all the abiding by all the laws, so that you will not make change in the human DNA or genome DNA. You can carry out the experimental work, but it should be a fruitful one, and it should not harm the human society or mankind. So recombinant DNA technology works by taking DNA from two different sources and combining that DNA into a single molecule. That alone, however, will not do much. So means if you are taking one DNA from one organism and the second DNA from another organism, and you are putting it in the first organism, then individually it is not playing an important role, but when you are splicing together these two different genes, then it will make a difference. So recombinant DNA technology only becomes useful when this artificially created DNA is reproduced. So why it is important? If you are taking the DNA from two different sources and putting it into the individual organism, but that in that particular individual organism, if the DNA is not replicating, if the inserted DNA, which is a chimeric DNA or the recombinant DNA, if it is not replicating, means it is of no use. So the recombinant DNA technology, it is becoming useful and fruitful only when this chimeric DNA is undergoing a replication process, or it is getting reproduced, and this is what is called as the DNA cloning or the gene cloning. So now we will see what are the different basic steps which are involved in the RDNA technique. So first, what you have to do, there are various simple steps, but each step has to be followed correctly. So first you have to isolate the DNA from any other organism. So here we have taken a eukaryotic cell. So this eukaryotic cell contains our gene of interest. Say for example, this gene is a pancreatic gene, and it is making a insulin molecule. For example, I am telling you, so this can be any eukaryotic cell, it can be a heart cell, it can be a kidney, or it can be a pancreatic, it can be a what we say, any other function, or any cell which is making that particular type of protein or enzyme molecule. So that DNA has been isolated now. So this is your DNA isolation step. So you have to isolate the first you have to isolate the nucleus, then you have to fragment out the DNA, and then you have to isolate the DNA in its pure form. Now this gene of interest, I have got it after DNA isolation. In the similar way, you have to, because the plasmid is a vehicle, or it is called as a cloning vehicle, or you call it as a vector, means who is taking or who is transferring the gene to the bacterial cell so that it can be expressed. It is the plasmids, or the cloning vehicles, or the vectors. So here the bacterium is having its own bacterial chromosome, and it is having a plasmid, and this plasmid is acting as a vehicle for transferring your DNA, into a particular host. So this plasmid, you have to isolate this plasmid from the bacterial cell. Then what you have to do after isolation of plasmid and after isolation of your gene of interest, you are inserting your gene of interest into the plasmid, and this is what is called as the recombinant DNA. This plasmid is called as the recombinant DNA. Why it is called? Because it is having a gene of interest from the eukaryotic cell, and it is a DNA from the bacterial cell, so it is a recombinant DNA. Now you can transfer this recombinant DNA via transformation processes. So the E. coli cell can take up this plasmids, and when this plasmid is taken up, usually you can transform this host cell. There are various methods, like if you want it to be in the eukaryotic cell, you can go for electroporation or shotgun method, or that we will be explaining you in detail in the next lectures. Or you can use protoplast fusion, or you can use a lysosome mediated fusions, etc., or polyethylene glycol fusion. So many more methods are available uh, for transferring this recombinant DNA into the eukaryotic cell. But in prokaryotic cell, it is a very easier method. Either you have to make this bacterial cell competent, so that this plasmid along with the gene of interest is taken up, or you can give chemical treatment like heat shock and calcium chloride treatment, so that the cell wall becomes more porous, and it will allow the entry of the plasmid, that is along with the recombinant DNA. Then later on, what happens, as the bacterial cell undergoes cell division, usually now this plasmid is also undergoing the reproduction or the cell division, and you will see the plasmid is containing the gene of interest. So you have got number of copies of genes. So this is what is called as the clone of cells, because from one cell, when it is undergoing a binary fission to produce daughter cells, it is called as the clone of cells. And usually this gene of interest is found in all the daughter cells. So this copies of genes have been usually have been found in all the clone of cells. Now what happens, these copies of genes can be extracted out. So when you are lysing these bacterial cells and then taking out this plasmid by plasmid isolation technique, alkaline lysis method, then you can take this, you can get your gene of interest in this way. Or you can allow the this plasmid DNA to undergo transcription and translation. Or so this vector, usually it is a transcribable vector. It is having all the genes responsible for undergoing the transcription, and because of this what happens, the messenger RNA is synthesized, and then messenger RNA is translated to give you copies of proteins. So these proteins, they can be used to make snow form at higher temperatures. So means at higher temperatures, if you want to make the snow, then these proteins will allow to make the snow. Or proteins can be used to dissolve the blood clots in heart attack patients. So when you are taking a heart attack therapy, usually certain proteins are responsible for dissolving the blood clots, and this can be usually done. Or you can see that copies of genes. Now this is transcribable and the translated product, that is the proteins. But if it is a gene only, then this gene can be transformed into the plants, and this gene can be insect pest resistance genes, or like bt toxin gene, which has been incorporated into the cotton plant or bt brinjal plant, so that this now bt toxin gene is producing the toxic product which controls the pest. Or this gene can be used to alter the bacteria for cleaning up the toxic material, or for cleaning up the petroleum or oil spills in the oceans, etc., or to clean up the xenobiotics or the aromatic hydrocarbons usually found when the oil spills are usually occurring in the because through the sea only, or through the oceans only, the oil barrels are moving from one country to another, because export is usually carried out by waterways and not from the landway or the airway. So this, it is polluting the oceans and the seas, and therefore you have to create certain microbes or genetically engineered bacteria are usually created so that they will clean up the toxic waste products. Okay. So recombinant DNA technology, we will see these are the 3 parts which are to be discussed in your syllabus. The basic concept of recombinant DNA technology, then what is the basic procedure of recombinant DNA technology, what are the different tools which are employed in the genetic engineering or the recombinant DNA technology, and what are the applications of recombinant DNA technology. So your both units, unit number 3 and unit number 4, is totally on the recombinant DNA technology, which deals on these 3 points, that is basic concepts, then basic procedures, what are the different tools employed, what are the vehicles used, what are the different steps used, and how the recombinant DNA technology is applied in the field of science, or what is the application of recombinant DNA technology. So first is the basic concept of recombinant DNA technology. So in the early 1970s, technologies for laboratory manipulation of nucleic acids was emerged, or just it started. In turn, these technologies led to the construction of DNA molecules composed of nucleotide sequences taken from different sources. So they started working with different DNA from different sources. The products of these innovations led to the recombinant DNA molecules, and this led to, this opened the exciting area or a exciting new avenue of investigation in molecular biology and genetics. Today we call it as recombinant DNA technology. So recombinant DNA combines the DNA from two sources. It is also called as gene cloning. So how it is helpful? Because we are creating a new combination of genetic material. And how it is helpful for human? Because earlier the insulin was obtained from cow, that is calf of cow. Pancreas was removed, and then from pancreatic islets of Langerhans, insulin was extracted out and then purified and then given to the humans for treating the diabetic patients, and this would result in the killing of the calf. So many more calves were killed in order to get just 1 mg or few mgs of insulin molecules. But now this insulin gene has been incorporated or integrated into the bacterial cell, and this human gene for insulin is now placed in the bacteria or the recombinant or genetically engineered bacteria. So bacteria are recombinant organisms, and now they started producing insulin in large quantities for diabetes. And because of this reason only, now the insulin rate has been declined. Earlier, the insulin rate for one vial, it was used to be 5,000 to 6,000, and only the rich people could afford buying this insulin, hormone insulin. But now it is easy to get, even at 500 rupees you can procure the insulin, that is insulin hormone. So genetically engineered drug is now available, and this first genetically engineered drug was obtained in 1986. So genetically modified organisms are also possible, and now we have seen what are the genetically modified organisms, because genetically modified organisms like bt brinjal plant is now producing bt brinjal, or you can see that the meat, uh, lean meat which is obtained from just growing the cells in the laboratory conditions. So it is playing a very important role for feeding the livestock also. So as well as it is sufficing the different needs of human beings also. So this universal, but you have to keep it in mind that it should not alter the DNA or the genome of the microorganisms or humans, so that it could lead into a creation of different type of say different lead to different type of diseases or uncurable genetic disorders. So it should not happen in such a way. And you know that in today's scenario, that is COVID-19, it is said to be that the COVID-19 is a genetically created virus particle in China, and it is devastating and is creating a havoc in throughout the world, and producing the uh, that is fatal respiratory disease. Okay, so genetically engineering is the application of technology to the manipulation of genes. So these advances were made possible by the method of amplification of any particular DNA segment. So how it was made possible? Earlier, they were knowing how to isolate the DNA, but how to make many more copies of that DNA was not possible. But with the help of now PCR or amplification techniques, now it is very easy to amplify that particular DNA several number of times. So whether it is regardless of the source or within the bacterial host, now it has become very easy because you can amplify that gene of interest in large amount, so that you can work with that particular gene of interest in a very rapid and suitable way. Or in the language of recombinant DNA technology, the cloning of virtually any DNA sequence has become feasible, because you can reproduce that DNA many more times with the help of different techniques. So recombinant technology begins with isolation of the gene of interest, that is, you call it as the target DNA, you call it as the foreign DNA, or you call it as the insert DNA. There are three names. The gene of interest is also called as foreign gene, or you call it as the target gene, or you can call it as the insert gene. This target gene is then inserted into the plasmid or a suitable vector, which can be a phage vector or the virus vector, or it can be a bacterial artificial chromosome or yeast artificial chromosome, or any shuttle vector, any of them, which we are going to see in detail. This recombinant DNA is then introduced into a host cell like E. coli bacterial cell, or you can introduce into the yeast cell like Saccharomyces cerevisiae, or you can introduce it in the cell lines. Different cell lines are produced for introducing this recombinant DNA. And then you can allow them to express the proteins, or you can just simply amplify your gene of interest or clone the gene of interest. This cloned recombinant DNA is then referred as recombinant DNA, and this procedure is called as the recombinant DNA technology. So cloning is very necessary. It is very important to produce numerous copies of the DNA. And since if you don't multiply this gene of interest, then your gene of interest may get lost while performing your experiment. So you need a number of copies of your gene of interest, and that is very important so that you can have a maximum amount of your gene of interest which can be inserted into the host cell. So RDNA technology, there are many different terms which are used for describing this technology. You can use the term gene manipulation, you can use recombinant DNA technology, you can use molecular cloning or gene cloning, or you can also use the genetic modification. So now there are certain terminologies which you should know. Cloning means, in a classical biology, a clone means it is a population of identical organism. Means, for example, if one bacteria is multiplying in a medium and producing the daughter cells or producing a population of cells, then you call it as a clone, which is derived only from a single parental organism. So this term in molecular biology also, the cloning of gene is derived from this bacterial cell population only. So it is a collection of molecules or the cells all identical to an original molecule or the cell. It means that it is a collection. Clone means it is a collection of molecules or the cells which are all identical to an original parent cell or the molecule. So recombinant DNA technology, a series of procedures are used to join together the DNA segments. Then this recombinant DNA molecule is constructed usually from two different sources, and then this, under certain conditions, this enter into a cell or is transformed into the either a bacterial cell or a yeast cell or a eukaryotic cell. Anything can do by means of different techniques being used, like electroporation, shotgun method, or by protoplast fusion, or by lysosome mediated, or any of these techniques you can use it, or by transformation technique you can use. So how the recombinant DNA technology works? Now these steps include, first, the isolating of the target gene and the vector. And then what you have to do, the first step is to isolate your target gene or the gene of interest. Also you have to isolate the vector, like the vectors like plasmid, or you have different genetically engineered plasmids like PBR322, POC19, or you have different lambda phage vectors, M13 filamentous phage as a vector, or you can have bacterial artificial chromosome, yeast artificial chromosome. Anything can be your vector. Then what usually, how the gene of interest is spliced or joined in the vector? It is by specific cutting of DNA. Specific cutting of DNA at defined site, and then joining or splicing of the DNA fragments. Okay, and then you are transforming this recombinant DNA into a suitable host cell. It can be a bacteria, it can be a yeast, or it can be any cell line. And after that, then you are looking for cloning or selecting how which of these bacterial cells are containing the recombinant sense. So you are doing the screening methods. There are many different screening methods which we'll be seeing in detail in the future lectures. So, and we will be selecting for the positive cells which contains the recombinant DNA. So selection, detection, screening, all these steps are to be carried out in order to find your recombinant DNA. And then you are either expressing this recombinant DNA in the form of large multiple number of gene of interest, or you can allow this recombinant DNA to undergo transcription and translation, which you call it as expressed at the end, or not in the end, or it cannot be. Means if only the gene of interest is undergoing only a multiple copies, then it is not in the expressed. It is not called as expressed. But if it is expressing itself, means it is making that particular type of protein molecule or enzyme molecule. Okay. So there are the 6 steps of recombinant DNA. What are the 3 steps? First is 6 steps. First is isolating vector and target gene. Then second is cutting or splicing the vector and the target gene. And then joining means ligating vector and the target gene together. And then fourth is the transforming, means transforming into a suitable host cell. That recombinant DNA is then getting inside the host cell, whether it can be a bacteria or it can be a yeast. Then undergoing a cloning, means you have to reproduce that recombinant DNA several number of times so that many copies are generated. And then you are selecting or screening the recombinant DNA containing bacterial cells or the yeast cells. Okay, so now we are coming to the second point, that is basic procedures of recombinant DNA technology, which we will see tomorrow. Okay.